Some scientific discoveries arrive with applause. Others arrive with skepticism, confusion, or even outright disbelief. And sometimes, a scientist can spend decades being ignored before the world finally realizes how far ahead of her time she truly was. Barbara McClintock belonged to that rare category. For much of her career, she saw things in genetics that almost nobody else could see. She uncovered mechanisms inside living organisms that challenged accepted scientific thinking. Many of her colleagues doubted her conclusions, not because her evidence was weak, but because her discoveries seemed too strange to fit existing theories. Years later, science would prove that she had been right. Her work transformed our understanding of genes, heredity, evolution, and the dynamic nature of life itself. What was once considered controversial eventually became one of the foundations of modern genetics. Barbara McClintock was born in Hartford, Connecticut in 1902. The world she entered was very different from the one we know today. Genetics itself was still a young science. Gregor Mendel's work on inheritance had only recently been rediscovered, and scientists were just beginning to understand how traits passed from one generation to the next. As a child, McClintock displayed an unusual independence. She enjoyed spending time alone, exploring nature, observing patterns, and thinking deeply about the world around her. Unlike many children who were content with simple answers, she seemed naturally drawn toward complexity. She wanted to understand not only what happened, but why it happened. This independence would become one of her defining traits. Throughout her life, she often preferred following evidence wherever it led, rather than conforming to prevailing opinions. This made her both an exceptional scientist and, at times, an outsider. Her family eventually moved to Brooklyn, New York, where she grew up. Although opportunities for women in science were expanding compared to previous generations, significant barriers still remained. Many people believed that scientific research was primarily a male profession. Women who pursued advanced scientific careers often faced skepticism, limited opportunities, and institutional discrimination. McClintock was not easily discouraged. She enrolled at Cornell University, one of the few institutions where women could receive serious scientific training. There she developed a fascination with biology, particularly genetics. At the time, scientists were beginning to understand that chromosomes played a central role in heredity. Chromosomes could be seen under microscopes during cell division, and researchers suspected they carried the instructions responsible for inherited traits. Yet much about their behavior remained mysterious. McClintock quickly distinguished herself through her ability to observe chromosomes with extraordinary precision. Many scientists looked through microscopes and saw structures. McClintock looked through microscopes and saw stories. She could identify subtle differences, trace chromosomal behavior through generations, and notice patterns that others overlooked. Her combination of patience, technical skill, and intuition made her exceptionally effective. By the 1920s, she had become deeply involved in studying maize, commonly known as corn. To many people, corn might seem like an ordinary crop. To geneticists, however, it was an ideal research organism. Different varieties displayed visible characteristics such as kernel color, shape, and texture, making it easier to track inherited traits. McClintock devoted herself to understanding the chromosomes inside maize cells. The work required incredible attention to detail. Long before computers or advanced imaging technologies, researchers relied heavily on direct observation. Hours were spent preparing samples, examining cells, drawing observations, and comparing results. McClintock excelled at this painstaking process. She developed techniques that allowed scientists to identify individual maize chromosomes with unprecedented accuracy. This achievement might sound technical, but it represented a major advance. For the first time, researchers could follow specific chromosomes and observe how genetic information moved between generations. Her work helped establish important connections between physical chromosomes and inherited traits. These findings strengthened the chromosome theory of inheritance, one of the central foundations of modern genetics. As her reputation grew, so did the significance of her discoveries. During the 1930s, she contributed to understanding a phenomenon known as crossing over. Scientists observed that chromosomes could exchange segments during reproduction, creating new combinations of genetic information. McClintock helped demonstrate how this process actually occurred. This was a crucial breakthrough because it explained one of the mechanisms responsible for genetic variation. Without variation, evolution would be impossible. The diversity observed within populations depended in part on these chromosomal exchanges. By her early 30s, McClintock was already recognized as one of the most talented geneticists in America. Yet her most important discoveries still lay ahead. The scientific community at the time generally viewed genes as relatively stable entities. Genes were thought to occupy fixed positions on chromosomes, passing information from generation to generation in predictable ways. This assumption seemed reasonable. After all, heredity depends on continuity. Traits remain recognizable because genetic information remains largely intact. But as McClintock continued studying maize, she began noticing strange patterns. Certain genetic traits appeared unexpectedly. Colors changed. Characteristics emerged or disappeared. Some regions of chromosomes seemed unusually active. At first glance, the observations were confusing. The data did not fit neatly into established theories. Many researchers might have dismissed such anomalies as experimental errors or statistical noise. McClintock did the opposite. She paid closer attention. The more she investigated, the more she suspected that genes might not be as fixed and stable as everyone believed. If she was correct, it would challenge some of the most basic assumptions in genetics. And over the coming years, Barbara McClintock would uncover evidence that life itself was far more dynamic, adaptable, and complex than scientists had ever imagined. As Barbara McClintock entered the 1940s, she had already established herself as one of the most skilled geneticists in the world. Her work on maize chromosomes had earned widespread respect among specialists, and her ability to observe genetic behavior under the microscope was legendary. Yet the discoveries that would define her career were only beginning to emerge. The more closely McClintock studied corn plants, the more she encountered results that seemed impossible according to conventional genetic theory. At the time, most scientists viewed genes as stable units arranged along chromosomes in fixed positions. A gene might mutate occasionally, but once established, it was generally expected to remain where it was. The chromosome was often imagined as a relatively orderly structure, carrying hereditary information from one generation to the next. McClintock's observations suggested something very different. She noticed unusual patterns in the kernels of maize. Some kernels displayed unexpected patches of color. Others showed traits that appeared and disappeared in ways that did not fit existing explanations. What made these patterns particularly intriguing was that they were not random. They seemed to follow hidden rules. Most researchers would have focused on confirming established theories. McClintock focused on the anomalies. She believed that nature often revealed its deepest secrets through exceptions rather than through ordinary cases. If something appeared strange, it deserved attention. This approach led her deeper into the behavior of chromosomes. Working at Cold Spring Harbor Laboratory, which would become her scientific home for much of her life, she conducted years of painstaking research. Day after day, she examined maize plants, tracked inheritance patterns, and compared microscopic observations with visible traits. Gradually, a remarkable picture began to emerge. McClintock identified specific genetic elements that appeared capable of influencing the activity of other genes. Even more astonishing, these elements seemed able to change their position within the chromosome. In other words, parts of the genetic material appear to move. This idea was revolutionary. Genes are supposed to occupy fixed locations. The possibility that genetic elements could jump from one place to another challenged one of the basic assumptions of genetics. McClintock referred to these as controlling elements because they appeared to regulate the expression of nearby genes. Their movement could activate certain traits, silence others, or create entirely new patterns of inheritance. Today, these elements are known as transposable elements or jumping genes. At the time, however, the concept sounded almost unbelievable. The evidence came from years of observation rather than a single dramatic experiment. McClintock tracked how specific chromosomal regions behaved across generations. She demonstrated that these mobile elements could influence the development of maize kernels, creating the distinctive pattern she observed. The implications were enormous. If genes could move, then the genome was not a static instruction manual. It was a dynamic system capable of rearrangement and adaptation. Genetic information could be regulated in ways scientists had barely begun to imagine. Yet explaining such a radical idea proved difficult. Science advances through evidence, but it also advances through frameworks. Researchers interpret data through existing theories. When a discovery challenges those theories too strongly, acceptance can be slow. This was exactly the situation McClintock faced. Many geneticists struggled to understand her findings. Some were skeptical. Others simply could not see how the observations fit into the models they were using. A few respected her technical expertise, but doubted her conclusions. The problem was not that her experiments were flawed. The problem was that she was decades ahead of the tools available to confirm her ideas. Modern molecular biology did not yet exist. DNA had not even been established as the primary carrier of genetic information. Scientists lacked the techniques necessary to directly observe many of the processes McClintock was inferring from her research. As a result, her work often received a cautious or confused response. Scientific meetings that should have celebrated her discoveries sometimes left her feeling isolated. She would present evidence that seemed compelling to her, only to encounter audiences uncertain about how to interpret it. This experience could have discouraged many researchers. For McClintock, it produced a different response. She became increasingly committed to following the evidence rather than seeking approval. If nature was showing her something unexpected, she believed her responsibility was to understand it as accurately as possible, regardless of whether others immediately agreed. This attitude reflected her unique relationship with science. Many scientists approach research as a process of testing hypotheses. McClintock often described her work differently. She spoke about listening to the organism, about immersing herself so completely in her subject that she could sense patterns others missed. To some colleagues, this language sounded unusual, but behind it was extraordinary observational skill. She spent so much time studying maize that she developed an almost intuitive understanding of its genetic behavior. This intuition was not mystical. It was built from years of meticulous attention, countless experiments, and an unmatched familiarity with her subject. By the early 1950s, McClintock had assembled a compelling case for transposable elements. She published her findings and presented them to the scientific community. The response remained mixed. A few researchers appreciated the significance of her work, but many remained unconvinced. Some considered the discoveries too complicated. Others thought the phenomena might be unique to maize and therefore of limited importance. As a result, the scientific impact of her research developed slowly. This was one of the most remarkable aspects of her story. Barbara McClintock had uncovered one of the fundamental mechanisms of genetics. Yet much of the scientific world was not ready to recognize it. Rather than fighting constantly for acceptance, she gradually shifted her focus. She continued working, continued observing, and continued exploring the mysteries of chromosomes. What she did not know was that future generations of scientists, armed with new technologies and new understanding, would eventually confirm her discoveries in dramatic fashion. The ideas that seemed so strange in the 1950s would one day become central to biology itself. But before that recognition arrived, Barbara McClintock would spend many years standing almost alone, trusting evidence that only she could fully see. As the 1950s turned into the 1960s, Barbara McClintock found herself in an unusual position. She was highly respected as a scientist, recognized for her technical brilliance and decades of important research. Yet the discovery she considered most significant remained largely unaccepted. Her work on transposable elements had not been disproven. Instead, it had been placed in a kind of scientific limbo. Many researchers simply did not know what to do with it. The world of genetics was changing rapidly. New technologies were emerging, and attention was shifting toward the molecular foundations of life. In 1953, James Watson and Francis Crick announced the structure of DNA, revealing the famous double helix. This breakthrough transformed biology almost overnight. Scientists suddenly had a powerful new framework for understanding heredity. Laboratories across the world rushed to explore DNA, genes, proteins, and the molecular mechanisms of life. Excitement focused on decoding the genetic instructions that governed living organisms. In this environment, McClintock's work seemed even more unusual. Most researchers were concentrating on how genes functioned within stable systems. McClintock was describing a genome that could reorganize itself, a genetic landscape that was dynamic rather than fixed. The scientific community was not yet prepared for such an idea. Rather than becoming frustrated, McClintock took a path that revealed much about her character. She continued working. She did not launch public campaigns demanding recognition. She did not spend years arguing with critics. Instead, she returned to what she loved most: observation, experimentation, and understanding nature on its own terms. At Cold Spring Harbor Laboratory, she maintained an active research program and became an influential figure for younger scientists. Many who met her were struck by her intellectual intensity. She approached biological systems with extraordinary focus, often noticing details others overlooked. What made McClintock different was not simply intelligence. It was patience. She was willing to spend years examining a problem without forcing it into a predetermined theory. She believed that nature often revealed itself gradually, and that researchers needed to remain open to unexpected possibilities. This philosophy shaped her scientific life. While others pursued fashionable topics, McClintock followed questions that genuinely fascinated her. She cared more about understanding biological truth than about competing for attention. Meanwhile, genetics continued advancing. As molecular biology developed, scientists gained tools capable of examining DNA directly. New techniques allowed researchers to identify genetic sequences, study mutations, and investigate the organization of chromosomes at increasingly detailed levels. Gradually, evidence began accumulating that supported ideas remarkably similar to those McClintock had proposed decades earlier. Researchers studying bacteria discovered genetic elements capable of moving within genomes. Scientists investigating viruses found mechanisms that inserted genetic material into host DNA. Other studies revealed that genomes were far more flexible and dynamic than previously believed. The picture that emerged looked strikingly familiar. Genes could move. Genetic elements could regulate other genes. Chromosomes were not static structures. Nature, it turned out, behaved much more like McClintock had suggested than many researchers had imagined. By the late 1960s and 1970s, the scientific world began re-evaluating her work. Papers that had once seemed obscure suddenly appeared remarkably insightful. Concepts that had been considered strange or overly complicated now fit naturally into emerging molecular frameworks. The discovery of transposable elements was no longer viewed as a curiosity limited to maize. It was becoming clear that mobile genetic elements existed throughout the biological world. They were found in plants. They were found in animals. They were found in microorganisms. In some species, they constituted significant portions of the entire genome. This realization transformed understanding of heredity and evolution. Genes were not simply passive instructions passed from one generation to the next. They existed within complex systems capable of change, interaction, regulation, and adaptation. The genome was alive with activity. For McClintock, this period brought a quiet form of vindication. She had spent decades trusting her observations despite skepticism. Now evidence from multiple fields was confirming the central ideas she had developed through years of work with maize chromosomes. Yet her response remained characteristically modest. She did not portray herself as a misunderstood genius finally proven right. Instead, she seemed genuinely pleased that science was moving toward a deeper understanding of nature. Recognition mattered less to her than truth. This attitude earned widespread respect. Scientists who interacted with McClintock often remarked on her independence of thought. She possessed a rare ability to pursue difficult questions without becoming distracted by scientific trends or external pressures. By the 1970s, younger generations of researchers increasingly viewed her as a pioneer. Many realized that her discoveries had anticipated major developments in molecular genetics by decades. What appeared radical in the 1940s now seemed prophetic. The significance extended beyond genetics itself. McClintock's work suggested that living systems were more adaptable and responsive than previously assumed. Genomes could reorganize, regulate themselves, and generate variation in ways that contributed to evolutionary change. Life was not merely following fixed instructions. It was interacting dynamically with its own genetic architecture. This idea would influence future research in genetics, evolution, developmental biology, and medicine. The scientific world was finally beginning to catch up with Barbara McClintock. And after decades of working in relative isolation, the recognition she had never actively sought was slowly approaching. The discoveries that once seemed impossible were becoming accepted facts. Soon, one of the longest-delayed acknowledgments in modern scientific history would arrive. By the late 1970s, Barbara McClintock had spent more than half a century studying genetics. Few scientists had devoted so much of their lives to a single field, and even fewer had maintained their intellectual independence for so long. The scientific world around her had changed dramatically since her student days at Cornell. Entire disciplines had emerged, technologies that once seemed unimaginable had become routine, and molecular biology had transformed the study of life. Most importantly, the evidence was now overwhelming. Researchers around the world were discovering transposable elements in organism after organism. What McClintock had first observed in maize was not an unusual exception. It was a fundamental feature of biology itself. Genes could move. Genomes could reorganize. Genetic systems were far more dynamic than scientists had once believed. As these findings accumulated, the importance of McClintock's work became impossible to ignore. Scientists who had previously struggled to understand her conclusions now recognized how far ahead of her time she had been. Her discoveries were no longer controversial. They were becoming essential. This shift was not sudden. It happened gradually as evidence emerged from laboratories across the world. One study confirmed part of her theory. Another revealed a similar mechanism in a different species. Then another. Piece by piece, the biological sciences assembled a picture that strongly supported the vision McClintock had developed decades earlier. The genome was not a static library. It was an active, changing system. This realization carried profound implications. Scientists began to understand that transposable elements could influence evolution by creating genetic variation. They could affect gene regulation, alter developmental processes, and contribute to adaptation. In some cases, they even played roles in disease. The very things that had once made McClintock's ideas seem strange were now recognized as important biological mechanisms. Her work was influencing fields far beyond maize genetics. Researchers studying evolution found that mobile genetic elements could help explain how species change over time. Molecular biologists discovered that genomes contained complex regulatory networks shaped in part by transposable elements. Medical scientists began investigating how disruptions involving these elements might contribute to certain disorders. The implications continued expanding. What had started with colored corn kernels was helping reshape humanity's understanding of life itself. Throughout this period, McClintock remained active at Cold Spring Harbor Laboratory. Although she was aging, her curiosity never disappeared. Younger scientists often visited her, seeking advice and insight. Many found her approach to science deeply inspiring. She viewed research differently than many of her contemporaries. For McClintock, science was not primarily about accumulating publications or winning professional competition. It was about developing a genuine relationship with the natural world. She believed that careful observation, patience, and openness were essential for understanding complex biological systems. This philosophy sometimes seemed old-fashioned in an era increasingly dominated by technology and specialization. Yet many researchers came to appreciate its wisdom. The more complicated biology became, the more valuable careful observation appeared. McClintock often spoke about the importance of paying attention to what organisms were telling researchers rather than forcing observations into preconceived frameworks. She encouraged scientists to remain humble before nature, recognizing that living systems frequently behaved in unexpected ways. This humility had guided her own work for decades. It had allowed her to notice patterns that others overlooked, and ultimately, it had led to discoveries that transformed genetics. Then came the recognition that many believed was long overdue. In 1983, Barbara McClintock was awarded the Nobel Prize in Physiology or Medicine. The announcement was historic. Unlike many Nobel Prizes in science, which are shared among multiple recipients, McClintock received the award alone. The Nobel Committee recognized her discovery of mobile genetic elements and the extraordinary impact that discovery had on biology. She was 81 years old. More than 30 years had passed since she first presented the work that formed the basis of the award. The delay itself became part of the story. Rarely had a scientific discovery taken so long to receive full recognition. Yet the passage of time also highlighted the strength of her achievement. The award was not based on temporary excitement or fashionable ideas. It reflected decades of accumulated evidence confirming that her insights were fundamentally correct. Scientists around the world celebrated the decision. Many saw it as a triumph not only for McClintock personally, but also for scientific perseverance. Her career demonstrated that important discoveries do not always receive immediate acceptance. Sometimes the evidence arrives years later. Sometimes entire generations of researchers must develop new tools before the significance of a discovery becomes clear. McClintock accepted the recognition with characteristic grace. She expressed gratitude, but remained focused on the science itself. Even after receiving the highest honor in her field, she continued speaking about the beauty and complexity of living systems rather than her own accomplishments. The Nobel Prize brought her name to a much wider audience. For younger scientists, she became a symbol of intellectual independence. For women in science, she represented a powerful example of achievement despite institutional barriers. For geneticists, she stood as one of the pioneers who helped reveal the true nature of the genome. Yet perhaps the most remarkable aspect of her story was that she had remained faithful to the evidence, even when almost nobody else understood what she was seeing. She had trusted nature more than consensus. And in the end, nature had proven her right. But the final chapter of Barbara McClintock's life would reveal an even deeper legacy, one that extended far beyond the Nobel Prize and into the very philosophy of scientific discovery itself. After receiving the Nobel Prize in 1983, Barbara McClintock became one of the most respected figures in modern biology. Yet what made her remarkable was not the award itself. It was the fact that the award had changed so little about her. She remained the same scientist she had always been, curious, patient, independent, deeply fascinated by the hidden patterns of life. Many people who achieve global recognition begin to focus on their legacy. McClintock seemed far more interested in continuing to learn. Even in her later years, she spoke with enthusiasm about genetics, evolution, and the endless complexity of living organisms. The Nobel Prize represented validation, but it did not represent an ending. For her, science was never about reaching a final destination. It was about understanding nature more deeply. As researchers continued exploring genomes during the 1980s and 1990s, the significance of McClintock's discoveries became even clearer. scientists found transposable elements throughout the biological world. They were present in bacteria, plants, insects, animals, and humans. In some species, these mobile genetic elements made up enormous portions of the genome. The implications were astonishing. What had once seemed like unusual genetic behavior in maize turned out to be a universal feature of life. Entire genomes contain sequences capable of moving, copying themselves, or influencing nearby genes. These elements helped shape evolutionary history on a massive scale. Many genetic innovations that appeared throughout evolution were connected, directly or indirectly, to the activity of transposable elements. The genome was no longer viewed as a static collection of instructions. Instead, it appeared more like a living ecosystem. Genes interacted with one another. Regulatory systems controlled expression. Mobile elements altered structures. Environmental influences affected outcomes. The complexity was far greater than early geneticists had imagined. In many ways, modern biology moved closer to the vision McClintock had developed decades earlier. She had sensed that genomes were dynamic long before molecular tools could prove it. Her observations revealed that living systems possessed flexibility, adaptability, and internal mechanisms that traditional theories had underestimated. Today, her discoveries influence fields ranging from evolutionary biology to medicine. Researchers investigating cancer study genetic changes that sometimes involve mobile DNA elements. Scientists examining developmental biology explore how gene regulation shapes the growth of organisms. Evolutionary biologists analyze how transposable elements contribute to diversity and adaptation over millions of years. Genetic engineers and biotechnology researchers work with systems that would have been impossible to understand without the foundations she helped establish. Her influence extends almost everywhere modern genetics is practiced. Yet perhaps her greatest contribution was philosophical rather than technical. Barbara McClintock demonstrated a different way of doing science. She showed that major discoveries often require patience rather than speed. They require observation rather than assumption. They require the courage to trust evidence, even when it conflicts with accepted ideas. Modern scientific culture often emphasizes competition, publication, and rapid results. McClintock's life offered a powerful reminder that understanding sometimes emerges slowly. Nature does not reveal its secrets according to human schedules. Some truths require decades of attention. Some discoveries require a scientist willing to stand alone. Her story also carries an important lesson about recognition. For many years, she received little credit for what would become one of the most important discoveries in genetics. If she had measured success only through awards, promotions, or approval from others, she might have abandoned her work. Instead, she focused on the evidence. That decision changed biology forever. When younger scientists asked about research, McClintock often encouraged them to develop a close relationship with their subject. She believed researchers should immerse themselves so completely in a problem that they could begin to perceive patterns invisible to casual observers. This approach helped her uncover truths that an entire scientific community initially missed. It was not magic. It was attention. Attention sustained over years, then decades. Barbara McClintock died in 1992 at the age of 90. She lived long enough to see the scientific world finally embrace ideas that had once seemed impossible. She witnessed the rise of molecular genetics, the confirmation of transposable elements, and the recognition of her life's work. Few scientists have experienced such a dramatic reversal of fortune. Fewer still have handled it with such grace. Today, her name stands among the giants of biology, not because she followed accepted wisdom, but because she questioned it. Not because she sought recognition, but because she sought understanding. Not because she worked within the boundaries of established thinking, but because she looked beyond them. In the end, Barbara McClintock transformed our understanding of heredity by revealing that genomes are not rigid blueprints, but dynamic systems capable of change, adaptation, and innovation. She taught the scientific world that life is more flexible, more creative, and more complex than it often appears. And she left behind a lesson that reaches far beyond genetics itself. Sometimes the most important discoveries are not the ones that fit comfortably within existing knowledge. They're the ones that force humanity to rethink what it thought it knew about the world.